In this video, we will discuss how insulin hormone is produced through genetic engineering techniques. So the first step is the isolation of gene of interest. First of all, the genetic engineer identifies the gene of interest in a donor organism. Special enzymes called restriction endonucleases are used to cut the identified gene from the total DNA of the donor organism. So, if we are studying the production of insulin hormone, so our gene of interest would be insulin from the chromosome of the donor organism. The second step is the insertion of gene of interest into a vector. A vector is selected for the transfer of isolated gene of interest to the host cell. The vector may be plasmid or bacteriophage. Now, what is plasmid? Plasmid is the extra chromosomal DNA that is present in many bacteria. And what is bacteriophage? Bacteriophage is a virus that infects bacteria. The gene of interest is attached with the vector DNA by using two types of enzymes. The one is endonuclease, that is breaking enzyme, and the other one is ligase, that is joining enzyme. The vector DNA and the attached gene of interest are collectively called recombinant DNA. The third step is the transfer of recombinant DNA into host organism. Recombinant DNA is transferred to the target host. In this way, host organism is transformed into a genetically modified organism. In this case, the host organism would be a bacterial cell. The fourth step is the growth of the GMO. The GMO means genetically modified organism are provided suitable culture medium for growth to give as much as copies of the gene of interest as needed. The fifth and last step is the expression of the gene. The GMO contains the gene of interest and manufactures the desired product which is isolated from culture medium. So in this case, the desired product would be insulin.